Good morning everyone. Today's topic is about stains. In this video we will see about what are stains, why should we use a stain, components of stain and types of stain. So the first question is, what are stains? Stains are colored organic compounds that are used for staining microorganisms. In short, stains are dyes that are used to give color to the microorganisms. Why should we use a stain? So when we observe a microorganism under a light microscope, they will appear colorless. In order to make them visible under a microscope, we give color to the microorganism so that we can study the size, shape and structure of that particular organisms. And the third is about components of a stain. There are three important components of a stain: benzene ring, chromophore and auxochrome. Together this benzene ring and chromophore are called as chromogen. So the first component is benzene ring. This is the structure of benzene ring. It is the colorless part of a dye and it is the basic structural component of a stain. And the second is chromophore. Chromo means color. So it is the functional group of the stain that gives color to the entire stain. And the third is about auxochrome. Auxochrome are nothing but group of atoms that are attached to the chromophore. They help in increasing the ability of the chromophore to observe more visible light. In short, auxochrome are attached to the chromophore and in turn they help in enhancing the color of the stain. Examples of auxochrome groups are hydroxyl group OH, amine group NH2, aldehyde group CHO and methyl mercaptan group SCH3. And the last segment is about types of stains. Stains contain salt which are made up of positive ions and negative ions. Depending on the types of stains, either positive ion or negative ion or both will be the chromophore. First we will see about acidic stains. So if the chromophore is the negative ion, then it is acidic stain. If the chromophore is the positive ion, then it is called basic stain. If both the cation and anion are colored part of a stain, then those stains are called neutral stains. Neutral stains are formed by combining both acidic stains and basic stains. Example for acidic stain is India ink, eosin. Example for basic stain is methylene blue and crystal violet. And examples of neutral stain is eosinate methylene blue and Giemsa stain. So the acidic dyes will stain the cytoplasm. The cytoplasm will have certain proteins and other components that are acidophilic in nature. Acidophilic means acid loving group. So these groups will bind to acidic stain. And basic stains are used in staining the nucleus and bacterial cells, because bacterial cell wall is negatively charged and nucleus is also negatively charged. So they will attract well to the positive ion, that is chromophore of this basic stain. And this neutral stain is used to stain both nuclei and cytoplasm because it has both the positive ion and negative ion or chromogen in the case of a neutral stain. And here the charge of the chromophore is negative. Here the chromophore is positively charged, but the net charge of the chromophore is neutral in case of neutral stain.